Descend into the Pacific off the coast of California, and the world drains of color. At the surface, sunlight blazes across the waves, but sink past 200 meters, and blue deepens toward black. By 600 meters, you enter a realm scientists call the mesophotic twilight zone, the border between the sunlit sea and the eternal dark below. Here, between roughly 600 and 800 meters down, only a whisper of light survives, filtering from above like the last glow of a dying evening. Nothing grows here. No plants, no reefs, only drifting particles, gelatinous animals, and hunters that have learned to wait. The pressure crushes. The temperature hovers near 4 degrees Celsius. This is one of the largest habitats on Earth, yet one of the least understood. And somewhere in this cold, silent column hangs a creature so bizarre that science got it wrong for more than half a century. Its name is Macropina microstoma, the barreleye fish. It is small, roughly 15 centimeters long, and for decades it existed mainly as a broken specimen in a jar, a puzzle no one could solve. Because everything researchers thought they knew about this animal was built on damage, the fish they studied had been torn from its world and destroyed on the way up. The living creature, the one that drifts patiently in the twilight, watching the faint shapes gliding overhead, remained a ghost. It possessed features so improbable that no textbook dared describe them accurately. A transparent shield over its skull, glowing green eyes that could pivot inside a dome of fluid, a face designed to deceive even the trained scientific eye. For 65 years, no human had ever seen it intact. The trawl nets that hauled it upward wrecked the very evidence that could reveal the truth. And so the barreleye kept it secret, suspended in the dark, hidden not by depth alone, but by our inability to observe it alive. To understand how profoundly science misjudged this fish, we have to return to the beginning, to a description written in 1939, based on a corpse that could never tell the whole story. In 1939, the barreleye fish entered the scientific record. Ichthyologists formally described Macropina microstoma from specimens dredged up by deep-sea trawls, nets dragged through the water column, hauling their catch hundreds of meters to the surface. What arrived on deck was a strange little fish with a small mouth, a compact body, and 2 upward-pointing tubular eyes. Those eyes gave it its name, barreleye, for their cylindrical, barrel-like shape. Researchers studied the corpses carefully. They noted the rigid green lenses aimed straight up, and they concluded the eyes were fixed, locked permanently toward the surface. This posed an immediate puzzle. If the eyes stared only upward, how could the fish ever see the food it was trying to eat? How could it aim its mouth at prey it could not look at? For decades, this contradiction sat unresolved in the literature. The prevailing assumption was that the barreleye's vision was simply limited, a specialized creature that spotted silhouettes above and somehow fed by luck or by tilting its whole body. Scientists sketched it with a solid, ordinary head. The face seemed simple. 2 dark spots appeared near the front, and many assumed these were the eyes, giving the animal a conventional appearance. Nearly everything about this reconstruction was wrong. The tubular eyes were not fixed, the dark spots were not eyes at all, and the head was anything but solid. But researchers had no way to know. They were working from evidence that had been mangled the moment it was captured. The nets that brought the fish up also stripped away its most crucial feature, a structure so delicate it never survived the journey. Generations of biologists examined dead barreleyes and confidently described an animal that did not exist. The truth was hiding in the missing pieces, in the parts that dissolved and tore before anyone could examine them. For 65 years, the textbooks preserved an error, and no one suspected how large that error truly was. The reason was heartbreakingly simple. Every barreleye pulled to the surface arrived with a piece of itself destroyed, a transparent dome that vanished before it could be seen. The barreleye's greatest secret was also its most fragile. Covering the top of its head sits a transparent, fluid-filled shield, a dome of clear tissue, soft and delicate, enclosing the entire upper skull. Inside this liquid-filled chamber, the eyes float, protected and suspended. It is one of the most extraordinary structures in the animal kingdom, and it is almost impossibly delicate. This is why science stayed blind for so long. When a trawl net swept a barreleye upward, the fish endured violence at every stage. The mesh scraped and battered its body. But the true damage came from the change in pressure. At 700 meters, the water presses down with enormous force. As the fish rose toward the surface, that pressure collapsed. Gases expanded, soft tissues ruptured, and the transparent dome, thin, membranous, filled with fluid, simply tore apart. By the time the specimen reached a laboratory bench, the shield was gone. What remained was a fish that looked decapitated of its most important feature. The eyes, no longer cushioned inside their liquid chamber, appeared as bare green tubes locked in place. The evidence had self-destructed on the way to the surface, erasing the very thing that would have explained the animal. Imagine trying to understand a diving helmet by studying only the head it once protected, after the glass had shattered and washed away. That was the barreleye problem. The fish did not hide its truth deliberately. Physics did it for them. Every capture destroyed the answer. And because it destroyed the answer consistently, every single time, for decades, scientists never even suspected the dome existed. There was no fragment left to hint at it. The self-erasing evidence produced a self-confirming error. Each new specimen agreed with the last, and each was equally incomplete. To see the barreleye truly, researchers could not bring it up to their world. They would have to go down into its world, into the crushing dark of 600 meters, and observe it alive, intact, with the fragile dome still whole. And in 2004, off the coast of Monterey Bay, robotic eyes finally did exactly that. The breakthrough came not from a net, but from a machine. In 2004, researchers at the Monterey Bay Aquarium Research Institute, MBARI, were operating remotely operated vehicles in the deep waters off Central California. These ROVs are robotic submersibles tethered to a ship by cable, carrying high-definition cameras and precise lights into a realm humans cannot easily reach. Piloted from the surface, they can hover at 600 or 800 meters, moving slowly through the twilight zone without the violence of a trawl. Marine biologists Bruce Robeson and Kim Reisenbichler were watching the video feed when something extraordinary drifted into view. There, suspended in the black water, hung a living barreleye fish, intact, undamaged, and unlike anything in the textbooks. For the first time in 65 years of study, human eyes were seeing this animal as it truly existed, and the difference was staggering. The fish's head was encased in a clear, glistening dome of fluid, the transparent shield that had never once survived a trip to the surface. Inside that dome, the tubular green eyes glowed, floating in their liquid chamber. The structure was perfectly intact because the ROV never lifted the fish out of its environment. The researchers observed it at depth, filming its behavior in its natural pressure and darkness. They watched it hover, nearly motionless, its whole shape revealed as it had never been before. Robeson and Reisenbichler carefully brought a specimen up alive, keeping it in cold conditions, and were astonished to find the dome could survive under the right care, confirming that the structure was real and not an artifact. The footage rewrote the science instantly. Decades of confident description dissolved before a few minutes of video. The barreleye was not a simple, upward-staring fish with fixed eyes. It was something far more sophisticated, a creature engineered for precision hunting, watching the water above through a living window. Technology had finally seen what physics had always destroyed. Now the real questions began. What were those glowing green eyes actually doing inside their crystal dome? And why did they burn with that strange, filtering color? Look closely at the living barreleye, and your gaze is drawn to two brilliant orbs suspended inside the transparent dome. These are the true eyes, tubular, elongated, and glowing a vivid, luminous green. Unlike the round eyes of most fish, these are shaped like barrels, giving the animal its name. And in their default position, they point almost straight up, staring through the clear shield toward the faint light filtering down from above. This upward gaze is the key to the barreleye's survival. In the twilight zone at 600 to 800 meters, almost no light remains. But what little does penetrate comes from directly overhead, sinking down in a dim, diffuse glow. Any animal swimming above the barreleye, a small fish, a drifting crustacean, a gelatinous creature, blocks that faint light and casts a shadow. To a predator staring upward, prey becomes a silhouette, a dark shape outlined against the last whisper of sun. The barreleye's tubular eyes are built to detect exactly this. Their elongated shape gathers as much of that scarce light as possible, and their upward orientation ensures they never miss a passing shadow. But the most striking feature is the color. The eyes shine with an intense green pigment, and this is no accident. The green pigment acts as a filter. In the deep sea, there are two kinds of light: the faint blue sunlight sinking from above, and the flashes of bioluminescence, living light produced by the creatures around it. Researchers believe the green filter helps the barreleye distinguish between the two, suppressing distracting bioluminescent glare so the fish can pick out the true silhouettes of prey against the downwelling light. It is a lens tuned to a single crucial task: reading shapes overhead in near total darkness. Suspended in fluid inside its protective dome, this pair of green tubes gives the barreleye a kind of vision no surface animal possesses, a specialized upward stare into a world of shadows. And yet, for decades, scientists thought the eyes were somewhere else entirely, because on the front of the fish's face sit two dark, eye-like spots that fooled generations of researchers into looking in exactly the wrong place. Look at a photograph of the barreleye, and your eyes go straight to the two dark spots on the front of its face. They sit exactly where a fish's eyes should sit. They are round, they are forward-facing, and for anyone glancing at a preserved specimen, they settle the question instantly. This is the animal's face, and this is where it looks. That conclusion is completely wrong. Those two dark spots are not eyes at all. They are nares, olfactory organs, the barreleye's equivalent of nostrils. Their job is to sample chemistry from the water, to detect the faint molecular traces of prey drifting through the black, not to form images. For decades, the misreading held because it made intuitive sense. A face with two eyes near the mouth matches every fish we have ever seen. The barreleye simply refused to follow the rule. The real eyes are the two glowing green spheres suspended higher inside the transparent dome, aimed almost straight up at the ceiling of the twilight zone. This split is the heart of the confusion. Sight and smell had been mapped onto the wrong parts of the body. Observers assumed the animal watched the world through the spots on its face, when in truth, those spots were tasting the water, while the actual eyes stared skyward through a fluid-filled window. Once MBARI's footage revealed the intact dome, the correction became unavoidable. The forward-facing organs were doing chemistry. The upward-facing tubes were doing vision. It was as if the barreleye had scattered its senses across its head in an arrangement no textbook had predicted. And this raised an obvious problem. If the eyes point straight up, fixed on silhouettes overhead, how does the fish ever aim its mouth, which sits far below and in front, at anything it wants to eat? A creature that watches the ceiling should never be able to bite what floats before it. For years, that geometry seemed like a fatal flaw in the animal's design. The answer would turn out to be stranger than a misplaced nostril, because the barreleye's eyes, it turned out, do not stay pointed up at all. The barreleye's eyes are not locked in place. That single discovery dissolved the geometric problem that had puzzled observers. When Bruce Robeson and Kim Reisenbichler studied the living fish, they realized the tubular green eyes could pivot inside the transparent dome, swinging from their default upward stare to a forward-facing position that lines the eyes up with the mouth. This is the crucial mechanism. In its resting posture, the barreleye watches the faint light above, scanning for the silhouettes of small prey passing overhead. But the moment the fish begins to rise toward a target, its eyes rotate. The green spheres tilt forward, tracking the very object they first detected as a shadow against the dim ceiling. By the time the fish closes in, its gaze and its mouth point the same direction. The transparent dome makes this possible. Because the eyes are suspended in a fluid-filled shield rather than fixed in open sockets, they have room to swivel freely while remaining protected. The window that once seemed like a fragile curiosity turns out to be functional architecture, a clear chamber that lets the eyes move without exposing them to the water. This resolved the paradox completely. A fish that appeared doomed to watch the ceiling while starving was actually executing a precise two-stage hunt. First, spot the prey overhead using upward vision optimized for faint light. Then, rotate the eyes forward to keep that prey locked in view during the approach and the strike. Detection and capture, handled by the same pair of eyes in two different orientations. It reframes everything about the animal. The upward stare is not a limitation, but a search mode. The forward swivel is not an accident, but a targeting system. Together, they let the barreleye convert a distant silhouette into a meal without ever losing sight of it. But knowing how the fish aims its eyes only sharpens the next question. What exactly is it aiming at? In a zone with almost nothing to eat, hanging in the cold and dark, the barreleye must find enough food to survive. And the way it does that involves a strategy stranger than a swiveling gaze. The twilight zone is a desert with water instead of sand. At 600 to 800 meters, food is scarce, sunlight is a whisper, and every calorie a fish spends must be earned back from an environment that offers almost nothing. The barreleye's answer is to do as little as possible. Most of the time, it simply hangs. Using slow, precise movements of its large fins, the barreleye hovers nearly motionless in the water column, holding its position while its upward eyes scan the dim ceiling for silhouettes. This stillness is a survival strategy. By refusing to chase and dart, the fish conserves the energy that constant swimming would burn, waiting instead for prey to drift into view above it. Its suspected diet points to one of the ocean's most dangerous neighbors. Scientists believe the barreleye targets small animals trapped along the tentacles of siphonophores, long, drifting colonial organisms, armed with stinging cells that stretch for meters through the dark. These tentacles snare tiny prey, and the barreleye may hover beneath them, watching, then rising to steal the catch. The transparent dome offers a compelling explanation for why this works. That fluid-filled shield may protect the barreleye's eyes from the siphonophore's stings, as it noses in among the tentacles to pluck away trapped food. The window that vanished in every net could be armor for a thief that raids one of the sea's most offended larders. It is a portrait of patience, a fish that barely moves, watching upward through a clear helmet, its green eyes fixed on faint shapes, ready to rotate them forward and rise the instant an opportunity appears. In a zone that punishes wasted effort, the barreleye survives by turning stillness into a weapon and transparency into a shield. Everything about it, the upward gaze, the swiveling eyes, the fragile dome, the hovering posture, fits together into a single, coherent way of life that took decades to see. And that is the lesson that lingers. For more than 60 years, the barreleye was described, named, and misunderstood. The truth was never hidden in some undiscovered trench. It was here all along, waiting for us to look correctly. Consider how long the error survived. Macropinna microstoma was named in 1939, described from crushed and torn specimens hauled up in nets. For 65 years, science held a confident picture of this fish. Fixed eyes, a simple face, no transparent dome. That picture was wrong in nearly every detail, and almost no one doubted it. The correction did not come from a bolder guess or a cleverer theory. It came from technology. In 2004, MBARI's remotely operated vehicles descended into the twilight zone off Monterey Bay and simply watched the barreleye alive, in its own water, at its own pressure. The intact dome appeared. The green eyes glowed. The face that lies revealed its true nature. What decades of trawling had destroyed, a robot's camera preserved. The lesson is humbling. The evidence had always been self-destructing on the way to the surface, and so the truth was mistaken for damage, and the damage was mistaken for the animal. We were not blind for lack of specimens. We were blind because we kept looking in the wrong way, in the wrong place, under the wrong conditions. Much remains unknown. We still have not directly filmed the barreleye stealing prey from a siphonophore. We do not fully understand how its eyes coordinate the swivel with the strike, or how many hours it can hang motionless, or how it finds mates in the vast dark. The Twilight Zone guards most of its secrets still, and every dive returns with more questions than answers. But the barreleye has already taught us something larger than its own anatomy. It stands as proof that certainty is not the same as accuracy, that we can name a thing, catalog it, teach it for generations, and still have missed what it truly is. Seeing clearly required not more confidence, but better tools, and the patience to look where the creature actually lived. The barreleye was never lost in some unreachable place. It floated in the near dark, watching upward through a window we could not imagine, exactly as it does today. For 65 years we simply failed to look correctly. The ocean hid the truth in plain sight.